Well, this is your opportunity to pitch your idea. Give us a clue about what you might have learned so far through your preliminary research and your literature reviewing. And also a time for you to garner hopefully some helpful feedback from those of us sitting up here, and hopefully Dr. Castangwe will be coming to join us. The whole idea that at this step and every other step along the way before you finish your thesis when you're seniors and cheering and get celebrating, every step of the way your product should improve. So you take every comment, every suggestion, you build that into a better product. And that's the way science and research works. You learn a little bit, you get critiqued a little bit, you make your next step a little bit better. So anything that's been will be brought to you is not a negative thing. It might have a negative tone, but it's a building block to be used to make your product better. That's where we're going. So this is the first chance you have to present what you put together to us who have not studied it intensely, and let us get an idea of where you're going and what we think some pitfalls might be and where we think you're headed that's very strong. And we were recording all of these, and we have your librarian here because you're in competition for the library award, an award from the libraries for the best presentation for the proposal. And the top 3 proposals will receive a financial award to help support the research. So money's on the line. Anything else? All right, so just give us your best shot.
Okay, so I'm Megan McGuffey.
Oh, I'm Anna Petnight.
I'm Asher Reddy.
I'm Anjo Choi.
I'm Jonathan Long.
Teacher Beck.
And our project is the study of pathology and potential therapy of a concussion. As you can see from our index, we'll be going through an overview of concussions, go through our literature review, go through our hypothesis and aims, and then outline our methodology. So the definition of a concussion is a blow to the head that causes the brain to shake inside the skull. Concussions are fairly common. It's an estimated 1.6 to 3.8 million that occur annually every year in the United States. One of the major problems with concussions is something called the secondary impact syndrome, which basically states that if you have a first concussion and are not fully healed from that first concussion and then have a secondary concussion, that second concussion will have much worse symptoms than the first. As you can see here, the symptoms are very different. For the first hit, it's headaches, memory loss, dizziness, fatigue, and nausea. However, for the second hit, it's amnesia, changing sleep patterns, sensitivity to sounds or light, respiratory arrest, and even coma. Also important to note is that for the first hit, the symptoms last 4 to 14 days, while for the second hit, they can last up to a month. So, something else important to note is that it's very difficult to diagnose the end of a concussion. So you might think that you're fully healed from your first hit. However, you could end up incurring a second hit with much worse symptoms without even really realizing that you were at risk for that. Okay, good. So when the brain is hit, the neuron cell membranes are stretched, causing ions into the neurons. These ions, did this work? Okay, sorry, that doesn't work. Letting ions into the neurons. This causes a lot of excitatory neurotransmitters to be released. One neurotransmitter, glutamate, in large quantities, repeatedly triggers a neurochemical cascade, which causes depolarization in the neurons. This depolarization allows calcium into the cells, which has a negative effect on mitochondria, and causes an increase in reactive oxygen species, ROS. ROS in turn causes a breakdown of lipids through oxidation, a process called lipid peroxidation, which causes the degradation of cell membranes. In order to repair itself, the brain needs a lot of energy. However, since the mitochondria are not functioning properly, the brain has to rely on the less effective anaerobic glycolysis, which ends up with the brain in an energy deficit. And now on to Ashwin to talk about the brain and glucose.
Now, just like virtually every other part of the body, the brain requires ATP to function. ATP is short for adenosine triphosphate. Now, to make ATP, the brain uses glucose through a process called cellular respiration. But during concussion, ATP production is actually disrupted, and it cannot do cellular respiration as well. So to offset this lack of ATP, the brain decides to increase the use of glycolysis, which is actually the first step in cellular respiration. However, the problem with glycolysis is that it does not produce ATP at nearly as efficient rate as normal cellular respiration does. So to offset this, the brain will pump more glucose, or the blood will pump more glucose into the brain to essentially make more ATP in a process called hypermetabolism. Now, the problem with hypermetabolism is that although usage will skyrocket over the first couple hours immediately following the impact, after a few hours, glucose metabolism will plummet because the brain can simply just not process that amount of glucose at once. It'll just essentially be overused. Now, that's unfortunate, but so there are a couple big glucose transport mechanisms in the brain. The two that we will be focusing on are GLUT1, which pumps glucose from the blood to the brain, and astrocytes, which are the green-looking things right here, and endothelial cells, which are the red ones right here. And the second glucose transport mechanism is GLUT3, which transports glucose into the neurons, which are represented on the diagram right here. In addition, we'll also be studying hexokinase, which is an enzyme that's used in the first step of glycolysis. What hexokinase does is it takes off the first one of the phosphates on ATP and makes it into ADP, adenosine diphosphate, and adds that phosphate onto glucose to make glucose 6-phosphate. And if you can go back one slide, as you can see right here, ATP is transformed to ADP, releasing 1-phosphate, which is added onto part of the glucose, making the molecule glucose 6-phosphate in the first step of glycolysis. And in addition, the concussion will also alter both the expression and location of these different transport mechanisms. Now, ROS, or reactive oxygen species for short, are a normal mitochondrial process. In a normally working brain, they'll be produced, but they will also be just eliminated at a healthy basal metabolic rate. However, during the concussion, ROS is actually overproduced, and the body cannot get rid of it in an efficient rate, so an imbalance will occur. This imbalance leads to many detrimental effects, such as a loss of ion gradients, which means the sodium-potassium pump is off and ATP is required to fix it, which, as I explained earlier, isn't always possible during a concussion. In addition, it also causes direct DNA damage, and it can be mutagenic, meaning it'll trigger mechanisms such as apoptosis and other cell-destroying mechanisms. But perhaps most importantly for our research, ROS will produce free radicals and peroxides, leading to a damaging of cell membranes in a process that's called lipid peroxidation. Now, in this diagram right here, as you can see, ROS can attack the cytochrome C complex, which is an electron accepting group in the cell membrane, which is heavily used in the electron transport chain, which is the third step of cellular respiration. Now, when ROS attacks the cytochrome C complex, it'll actually leave the mitochondrial membrane, which doesn't allow for the electron transport chain, and therefore cellular respiration to work nearly as efficiently, causing the decrease in glucose that we see, or decrease in ATP that we see during concussion. So, just as an outcome of concussions, as I explained earlier, lipid peroxidation is a large effect due to ROS. In addition, we see the neuron itself, more specifically the axon, stretching out, which causes disruption of the sodium-potassium pump, and which also leads to an influx of calcium ions, which will lead to an uncontrolled release of neurotransmitters. In addition, ROS will increase the amount of oxidative stress in the cell, which can overwhelm it and will be very difficult to get rid of. Now, one thing that we did find in our research is that vitamin E, which is a very commonly known antioxidant, has actually been shown to reduce lipid peroxidation that's been found to occur through ROS. What it does is that it will react with the ROS, and it'll produce a non-lethal substance, like a disposable substance that can just very easily be gotten rid of. And this graphic right here is it's a resting state fMRI, which really shows consistent changes across the brain pathophysiology. The left image shows a completely normal, uninjured brain. whereas the right image shows a brain that's been concussed actually 10 days after the fact. And as you can tell, you can see the differences in colors showing that even 10 days after concussion, which is a very long period of time, brain activity is still pretty significantly affected. So obviously this is a pretty major problem. And just quickly, I want to go over the current research in the field right now. As I explained earlier, antioxidants has been shown to reduce ROS, and ROS is also increasing during a concussion. Now, a TBI, or a traumatic brain injury, has been shown to increase the expression of GLUT1 and GLUT3, which are the two main glucose receptors I explained earlier. However, a mild traumatic brain injury, an MTBI, or actually as is commonly known, a concussion, have not been studied nearly as much in the current literature today. And in addition, the interplay between GLUT1 and GLUT3, hexokinase, lipid peroxidation, and ROS in a mild traumatic brain injury has not been studied at all. And this is the niche that we want to fulfill with our project. In addition, most of the studies, if not all of them actually, that look at a traumatic brain injury have been done in vivo, meaning that they've either been done as case studies on humans who have suffered TBI or have been done on rats or mice or the like. Our experiment, as we explain later, will be done on an in vitro cell model. So we can actually examine the interplay between all the factors that I just explained earlier. Now, this has not been done nearly as much in the current research, so this is something that we hope to discover.
Okay, so onto our hypothesis. Basically, concussions cause many chemical and physiological changes in the brain. So basically, these consequences include changes in glucose transport mechanisms, as well as an increase in the production of ROS, as stated earlier. And in our model, we'd like to see how these changes affect our endothelial cells, our astrocytes, and our neurons. And we also want to see the correlation between these factors, as well as how antioxidants, specifically vitamin E, may have an effect on these changes. So we'll look at these, our model, before or after our injury, as well as after protecting it with vitamin E. And we'll be taking three main aims into account. Our first aim is to look at elements involved in glucose transport, GLUT1, GLUT3, and hexokinase. Our second aim is to look at oxidative stress levels, and to do that, we'll measure the amount of reactive oxygen species in our models, and look at lipid peroxidation. Our third aim is to look at overall cell damage. And to that, we'll use cell viability tests, and also measure the extent of neurite outgrowth in our model. So we have four experimental groups that we'll look at. Our control is just growing healthy cells. We have to establish a baseline. Our second group will have one concussion. Our third group will establish a baseline for a second impact syndrome. So we'll apply one injury and then apply a second injury afterwards. Our fourth group, we will test the effect of vitamin E. So we'll apply an injury first, apply the vitamin E, and then apply the second injury. And of course, we'll be taking our parameters into account. Because of uncertainty of when cells start indicating concussion cascade damage, we'll be testing these at different times. Right now, our tentative times are 1 hour, 5 hours, and 24 hours after we establish our initial conditions. So this is the model that we'll be using. In the top, we have the removable transwell, and we'll grow our endothelial cells in the top portion. On the bottom of the transwell, we'll grow our astrocytes. And this will simulate our blood-brain barrier. In the bottom part of the well, we'll have our neurons. So, as stated earlier, we'll need to stretch axons in order to mimic the concussion. And in order to do this, we have a device, we'll use a device called the cell injury controller. And basically this apparatus delivers a pulse of air into the well. And to do this, we'll remove the transwell part and leaving only the neurons on the bottom well. And we'll be using a special type of well that has an elastic membrane on the bottom. So when the device blasts air into the neurons, it'll be able to stretch, and we'll be able to measure that stretch.
Now, in regards to our antioxidant treatment, we want to use the alpha-tocopherol form of vitamin E. This is one of the most inexpensive and commonly found forms of vitamin E. And it's also capable of crossing the blood-brain barrier, so it's very useful to our research project. And we'll be inserting vitamin E through the top portion, where it can reach the endothelial cells and astrocytes, as well as the bottom portion, which will reach the neurons. And we'll use a concentration of 1 millimolar. And the vitamin E will remain there during our experiments. Now, when we're looking at elements of glucose transport, we want to see their expression level as well as the location in each cell type. So to do this, we'll be using Western blot, using first and secondary antibodies of cell lysates, and use densitometry to analyze how much of each element of glucose transport is expressed. Now, to find out where each element is, we'll use fluorescent microscopy, and as you can see here, we've designated the colors of dyes that will be visible for each element. And I'd like to point out that this timeline is how long we expect to take for each of our experiments in the following slides as well. Now, when looking at oxidative stress, we want to measure ROS levels as well as lipid peroxidation. Now for ROS, we plan on using a CellRox reagent, which will bind with ROS and turn it a green color, as you can see in this image on the right, far right. Now for lipid peroxidation, we've determined that F2 isoprostanes are a product of peroxidation and can serve as a measure of the cell damage caused by lipid peroxidation. We'll be using the antibodies for the F2 isoprostane, and this will come up as a green fluorescent color, which we'll analyze through microscopy. Now, in order to measure neurite outgrowth, we'll be using microscopy again, only this time we'll use a crystal violet dye, which will come up as purple, and we'll be able to see how far the neurites have stretched out. Now, in order to determine cell viability, we'll use a 2-color live dead assay kit. As you can see here, green cells will represent live and healthy cells, whereas the red will represent dead and damaged cells. Now for our analysis, we'll repeat each experiment 3 times and using 2 separate wells for each experiment. And when we extract cells for analysis, we'll take 20 from each experiment, giving us a total of 120. Now we'll be using a t-test to compare between 2 different groups. For example, healthy versus concussed cells, or 1 concussion versus 2 concussions. And the ANOVA test will allow us to compare between multiple groups at a time. In case our predetermined plans do not work out, we have several alternatives listed here, and we may use them depending on our experimental success. And we hope to fill in any gaps in knowledge with our research. As Ashwin mentioned, there's not much research done on how each cell type interplays with each other in regards to concussive damage. And we want to see if vitamin E can act as a protection method rather than as a therapy for repeated concussions.
All right, well, very good. We typically start off questions with our expert, but Dr. Steinway does not have any questions right now. So what we'll do is just open up to the rest of us to ask questions there. I have a couple comments and questions, then we'll kick it around the room. First, I really liked in your written proposal, when you showed your aims and aim 1.1 and 1.2, 2.1, 2.2, that you had, this is the injury and this is the protection, and and to me that was brilliant. I love that, how it looked to me on the page. And in your slide, you left it off. I was like, I actually noted, I said, this is very nice, but I wrote it on the paper. That's just a great way to present it, so don't cut yourself short, put it back on there. Just a comment. It's like your Transwell device, okay? I know you're basing a lot of your research on that device working properly. In reading your proposal, I know you reference that it's been used. I didn't get a confidence level of how good of a model it is, how well it works. What do you think? Is it good? Does everybody use it? Is the standard, or is it something new and novel? You're putting all your eggs in a basket that's a solid basket, or are your eggs in a basket that's a brand new basket? I need some comfort level that this tool is going to work.
Well,
Convince me.
we know the researchers at VCU have created and tested this injury device.
The Transwell.
Oh, I'm sorry.
Well,
The injury device, I think, is simple enough. You blow air on it, stretch it, that should work.
well, the injury device, or not the injury device, sorry, the Transwell model actually has been shown to,
Yeah.
I can't pull the paper right now, but it's actually been used to mimic the blood-brain barrier, which is a lot of our experiments based on that.
Mhm.
What we're doing that's pretty new is adding the element of stretching the neurons and then putting them in the Transwell.
Right, but is the Transwell published multiple places, or is it just
Yeah.
one
It's
lab?
widely
that uses it?
used, not only in the
It's-
blood-brain barrier, but they use it to mimic all kinds of membranes.
Okay,
So,
that's good. That's what I need to know. I think knowing that most of the readers of your product you generate are going to be more of the lay reader than the very experienced reader. You need to convince that this is a good approach. This is a tried and true. It's simple. We should do this. This should work. This is not where we should have concerns or doubts. Now we'll go back to the high-tech air compressor. Okay, that logically makes a lot of sense to me. I mean, it just makes sense that this should work. Even though it may not be widely used, it's novel and it just makes sense that it should work. And the first couple times you do it, you're gonna blow the bottom out of the thing because the air pressure turned up too high. That's to be expected, right? So you'll make sure you have more than one set up to use. But I'm convinced that will work because it just makes sense logically. The Transwell, I didn't have that feel for whether I was comfortable with it or not. But if it's tried and true, great. Okay. Anyone else want to go with a question, comment?
I think it's a pretty fair concern. I mean, these Transwells, and this is something that I think you guys, need to get as well, but, so, these Transwells have been utilized widely in the
Okay.
literature. Typically to mimic a barrier that is semi-permeable so that substances in one timber do not simply passively leak into the other or by diffusion, etc., right? And the big concern with them is precisely that they do not establish good enough barriers.
Mm-hmm.
And so if the goal of utilizing them is to independize compartments,
Mhm.
truly, then there is a concern.
Okay.
There is a concern. Now, in their project, there is nothing that involves a true separation. We just mean to have all the three cell types in the same environment so that it can be a crosstalk between all the cells involved in the process. That's basically what we want, so.
So when you introduce the vitamin E, I'm probably oversimplifying, but you drip it in the top. It will diffuse and be transported across the barrier into all the above and below the barrier.
Well, actually, we're putting it in both compartments so that diffusing across the membrane isn't really an issue.
Okay.
It might diffuse, it might not,
We just want to ensure that the vitamin E will reach every cell type. So
yeah.
yeah.
we'll take the top insert out and apply it to the neurons, and then we'll put it on the top insert, which will get to the other two cell types.
Right.
Yeah, the main point is just to see whether it can be used as a preventive mechanism for the second injury. So we're not really concerned with drug delivery right now or getting through the membrane.
Right, okay. Good.
I don't have anything to add to what they are doing as a project, you know, because I don't understand much about it. I'm not in the sports either, so, but I can speak about the library portion of your project. Because you had so many citations in your presentation, I wish you had a list of the cited references that you had listed, right, during the presentation. Along the lines, I don't know if you know, because I looked at your paper, and all these images are also in the paper. And you say it's coming from this source. Images are not the same way copyrighted as the rest of the journal articles. So just by stating where it's coming from is not gonna make it. Because these will be published eventually in DRUM, which is the digital repository of the University of Maryland, and it will be crawled by Google. And if you had reproduced an image without permission, that is a no-no. So you can say reproduce with permission, for example, or something like that. And probably they will grant you the permission. It's not like they won't grant you the permission, but you should be aware of that. For this part of the project is fine, but I'm just a word of caution for the future. And something else that I noticed also in the references, you're citing some references that are not, that I've never heard of. For example, medicinenet.com. I don't know what it is. They have, it's a popular site, it has a lot of advertisement. They have a seal of approval that expired May 2013. So I don't think you should be referencing them. You can use the information from there, but try to find that same information in a place where you can actually cite, not this particular website. The same goes for one that is called News, something about medical news today, which usually when a source, like a news source, they are citing, they are saying where they got the information from. So instead of citing that particular one, you can go to the places that they had used to produce the news. I don't know if I'm making myself...
Yeah, go to the original source. Yeah.
The original source. And I didn't go through all the references, I just noticed the few. And I'm going to go back again to the same, to the paper. And this is formatting. For this purpose, probably it doesn't really matter, but you start with reference 4 instead of reference 1. I mean, it's probably when merging the documents somehow, these softwares do wonders for us, but also create a lot of anxiety. So for your final product, try to start with number 1, not number 4.
Okay.
And building off of that, how did you, let me get my exact question. How did you explore, assess, and integrate scholarly resources into your proposal writing process?
Well, I mainly search through databases using just keywords at first. Like, you know, you may start with traumatic brain injury, and then when you find an article, you pick up on keywords, you know, like the glucose transporters or reactive oxygen species, and then you search for narrow things. So.
Like I remember when I first started the project, I had no idea what GLUT1 and GLUT3 even did. Then I just frankly went in the database and searched up concussion. And then I found a little bit about GLUT1 and GLUT3 is how they worked in glucose transport. But from there, search GLUT1 and GLUT3 and concussion. And then, narrow it down to and traumatic brain injury. Just keep going, bringing narrower and narrower scope.
Well, based on my experience, yeah, like Ashton said, we didn't know much about this at all. So I think a lot of the things we did were just reading, not exactly citing all the sources that we read, but we just did a whole lot of reading from scholarly databases, and that gave us like a base knowledge that we used to search for more specific articles.
So which database did you use? You say scholarly databases. Which one?
We used PubMed, and I used a lot of Google Scholar.
Mhm.
Used academic search.
Okay.
I know Science Direct for me was a big one.
Yes, Scopus and Science Direct, yeah, that's a good one for this type of thing.
And make sure you use your resources too. There's, you can visit Navenka in the EPSL library. And also, I think your advice about digging through some of those sites. I didn't see any Wikipedia references, which is good. But some of the websites, you do have to be careful, because it's not all scholarly information. Some of it is there for advocacy reasons, which is all well and good.
Yeah.
But you need to be able to dig through to find the research that's informing those papers.
And I wanted to say as well, because this is also a very popular topic, and the audience is the public. So using these popular websites is not a bad thing, but maybe just reference it. The popular websites state this, so you have something, that this is a popular website, but you still wanna cite it because that's where the public is getting the information.
Great. Can you run back to the slide you had of the side-by-side MRI images? Okay, this is not to put you on the spot, but, so I'm consuming this information, right? You just laid it out on the table for me to look at. What am I to assume about those two scans?
Well, like I explained, the first scan is just an uninjured brain, so not concussed at all. And then the second brain is one that suffered a concussion, and then this is the resting state fMRI 10 days after the fact. And I should have explained this, but
Let me just ask you, fundamental question, is that the same brain?
yes, or actually,
To me, that's very important. Is that the same person or not?
to be honest with you, I'm not actually sure.
Because if you scan my brain and scan Dr. Murrow's brain, I'm sure hers would look a lot more brighter. but they would look different. I mean, the indicators, they would look, those look identical except for the color spots. I mean, to be different, they're the same size. I mean, shapes the same, or am I reading too much into it? I mean, I really want to know as a consumer. Okay, and this is why you have to learn to get your message across to the consumer. I'm consuming this information, and I want to know, is that the same person? And is it a person that had a baseline injury, and I'll tell you why I'm asking this question in a moment. A baseline injury, a baseline image, and then happened to be concussed later and had an after image. Or did someone just predict they're going to get a concussion, so they said we'll get a scan and then we'll wait for the concussion to happen? So, or is it the two different people? Because if it's two different people, I don't know if you have a good control, and I don't know if I believe your message.
I would, again, I'm not 100% certain, but I would honestly lean towards the former, like it was a baseline for like perhaps an athlete, and then maybe they were playing football and then they had like a concussion, and then maybe this is the 10 days after slide. Again, I'm not 100% sure, but that's just what I would be inclined to
Okay.
believe.
That's the kind of thing you should probably be aware, you should find that out. Because next time you present, you won't be able to answer that question. Because that's something that jumped out of me out of the question. And the reason I asked is that my son is a college football player, and he got pre-baseline scan done. And thankfully, he's done now. And has never had to have the post scan done. So he's part of a study. I just wondered, because I have the DVD at home, and I can watch his brain. But I would really want to know, when you come to your thesis presentation in a couple years' time, and you're going to use this kind of data, I want to know if that's the same person or not. I might not follow all the other language you're using and all the other slides, but that's something that, as an audience, a consumer, I want to know. Okay. Simple as that.
And that's another example too, because I just pulled up the article based on your reference. You can also dig in and go to the scholar's work. It says who it is and where that person's from. So it's a researcher at Penn State. You probably could dive in to find more from this particular study. This is just a snippet that the NIH used for a press release, which is good. That's getting the information out. But it's another example of just digging a little bit deeper into the information to provide support for the story that you're telling.
Okay.
I have another question,
Go ahead.
and so I'm a little more team-based, but your budget is rather sizable.
That's where I was going.
I know that it is very costly. How do you plan to fund your project? And especially if you want to do something with 4 and 6 months, you need to start soon. So what's your plan?
So we actually just formed a funding team this month, and we found several sources that we can apply to. And so we're going to start applying to those. I think the first deadline's in March 1st.
Yeah, it's March 1st.
Okay, but funding deadline, if a funding agency deadline's March 1st, when do you have to have your proposal ready?
A lot before.
Roughly a month,
Aim for a month ahead of time, which is last week. Right?
yeah. Right.
Okay, it takes at least 2 weeks to get it off campus and so that she's happy with it and everyone signs off on it and so it gets off campus. But just a rule of thumb, have it ready a month ahead of time and you'll be better shape. Yeah, March 1st is already pushing the limit for March 1. Okay? Great question.
And that's something too where I think my observation, just even getting us set up for today, we need to do some more planning ahead for the team, right? And work on some communication. So whoever the funding team is, come in, talk to me, talk to Dr. Cole, make sure you're talking to your mentor, and let's come up with a plan. Because HHMI and ACC IAC are the last round of funding. Those are coming up in April. That's internal funding, so that's good because it means there's no overhead. They don't take out any percentages. That's direct money to you all. But it's not going to be $26,000. So coming up with a plan to fund your project, but also how are you all going to hold each other accountable and make sure that communication lines are flowing well for your team.
That's pretty important because as it gets, the intensity is going to ramp up now. I mean, you think you've done a good bit of work to this point? You haven't done anything yet. It's going to really ramp up, and knowing what the right hand, knowing what the left hand is doing is going to be even more and more important as you go along. Back to the funding issue. Okay, what's Plan B? You don't. I mean, $26,000 doesn't come to you. What are you going to do?
I know, oh, go ahead.
Oh, okay, we're probably not gonna deal with the vitamin E treatment, we'll just deal with, trying to just see the interplay of the 3 cell types, because that's still pretty significant.
Okay. So which aim was the vitamin E? Is that aim 3?
Yeah, this is the 0.2s on all of them.
Oh, the 0.2 on all that, right, got it. Okay, so I can't look at your budget, I can't figure out what that would save you by looking at your budget.
It would take out a number of the cells.
Yeah.
Like right now, our budget's calculated for the amount of cells and media we need for
Uh-huh.
all of the aims.
Right?
So that would take out an entire experimental group,
Right, so,
which is a fourth of the.
yeah, so about 25, cut by, about 20%, maybe, that's my fair estimate.
Okay.
So that's still, I don't know.
20,000.
Yeah, $20,000, right? So, what's the plan? What's the plan C?
That's what
We're not
it's-
discouraging you, we're just trying to make sure you realize how important this is.
And we've also been looking at some unorthodox funding grants, like I know Charlie and I have been in contact with the agent for Darius Webb, who's actually plays cornerback for the Baltimore Ravens. So we're actually looking into the foundations for like NFL players and to see if they can maybe sign off on like a small donation, because it'd be great press for them and for the NFL because concussions are a very hot button issue.
There's a research team at Union Memorial Hospital MedStar in Baltimore that's focused on football players and concussions. There's a guy up there named Andy, I don't even know what Andy's last name is. It'd be worth a phone call or to have a conversation saying this is what you're into. I mean, they're looking for funding too, but this is what I'm into. We're looking for $25,000 to do this project. Can you give me some tips where to look? We're not competing with you. We're just a bunch of poor little college students trying to do a project. Chase down leads like that. Your idea about going to the NFL is fantastic. I mean, that's chump change for those guys, right?
Right.
So go for it. I mean, that's very good stuff. But remember to keep all that very, very professional. I mean, if someone wants to make a gift, to your team, come talk to us and figure out how to do that, okay? We have to make sure it's done totally professionally and properly, but we can do it. Okay, we can do it, we can take a check from anyone, we just handle it properly. Okay, that's
Almost
great.
anyone.
Almost. Yeah, almost anyone. Okay, so that's great, but your budget is more, your allocation we're gonna give you per year is not gonna cover this.
Right.
Okay. So let us know how you're proceeding there, and let us know if we need to help you along those regards. Okay, one of the different non-money issue is, you mentioned a lot of different trainings and certifications that the team's gonna have to get before you can start something. Lab safety to start out with, and all the other, there's a whole list you listed of different things. How are you progressing on getting those accomplished?
I believe all of us actually completed the new researcher training, which is actually a real seminar. As far as I think most of the other ones are actually online training, like I know for a fact I've completed all of them myself. It's not too bad actually, so we could actually probably have that done. We have a goal by the end of the month for our entire team to finish that, so.
And the goal is to have every member certified or trained in every required area.
Yes.
Okay. Hold each other accountable on that, make sure there's not a straggler. Okay, on your back. All right. Overall, I thought your proposal was very good. I was very impressed with it. That was very good. I like the embedded images, just make sure they're properly credited, or else that will hold up your thesis publication. Okay? You don't want that to happen when you're ready to leave here. But I think it was very well written. You guys have come a long way since I talked to you last spring about this project. I think you've taken a big idea and boiled it down to an answerable series of questions, which, as long as those little boxes work, and the little air pump works, as long as that hardware works, and you don't blow everything up with the air pressure, that's just trial and error. Take some advice, talk to some people about how to do initial settings before you break everything. It sounds like very well thought out, very orchestrated series of questions to be answered on a scale that you can handle. So I think it's a good project. I just hope you are able to fund it and get it all done. Anything else? Any questions, comments from you?
Not really. I mean, I think you guys have touched upon most of my concerns. I'll take the blame for taking out the very last slide with the list of references, because that was me telling them we never show this, so take it away. So I take the blame for that one. But no, I mean, I think the funding issue is probably the most concerning one, because if you start thinking about it, as you've been pointing out to, right, what you need to cut if you don't get the fund is the entire project, right? So I mean, you can cut the vitamin E, but that's only 25%. Let's say you only are able to get $8,000. What part of your project are you able to do with that? I mean, what are gonna be your priorities? Let's put yourself in that position. You get $8,000. So from your list of 4 different groups and different items that you're gonna be evaluating, what are your priorities? Imagine that you just have that amount of money. Where do you start?
Just do one concussion and then just see the interplay between the 3. Because it's still pretty new. There hasn't been much work on that. So it would be pretty far downsizing. We don't get to say all the crazy stuff that happens with secondary concussions, but concussions are still a really major problem.
Mhm.
And yeah, that would cut a lot of it.
I was thinking, alternatively, we could actually just focus on one of the elements. We go back to the literature and just take a look at which one has the least amount already published, and then just go with that and hope we fill a niche that hasn't been published yet.
Yeah, I mean, we're not trying to be discouraging at all. We're just trying to let you think about, this is something we do on a daily basis in laboratories. You say, God, I wish I could do that, but for some reason or another I can't. So you cut it back a notch. You gotta figure out what your plans can be. Right now, just aggressively go after funding, see what you can find out. And maybe we'll be able to do it all.
Yeah,
my other question is about your organization. I know that I have asked you to think about this, and it's not very easy to do one, I mean, before you get into the lab and you see the real situation there. But you have a lot of things and a lot of, groups and a lot of markers you're gonna look at. Do you guys have an idea of how you're gonna organize yourselves? Who is gonna go after what? Are you gonna form teams for looking at particular aims or looking at particular methods, or?
Our main idea was to have people divide up into groups for each particular aim and learn each of the two. There are two main methods for each aim, and so then those people will be experts on each of the aims. We haven't assigned people to them yet, but we will do that after we get into the lab and get a feel for what people are used to.
I would just advise and have you think about ahead of time, how will you even schedule people to get in the lab? How will you hold each other accountable? And how will you increase your communication? Because I think from today, let's use today as a learning experience, Hanjo was running here from class, we needed a backup laptop, did Charlie contact the person? Making sure everyone's on the same page will be especially important in the lab, because if I run one test on our very expensive cells and then Ashwin comes in and runs the same exact test on the same exact cells, now we have a problem. So you don't want to do that. You want to make sure that you're keeping good lab notes, and that, if I'm scheduled to come in the lab at this time, I come at that time, because if I come later, then now I've messed up for Megan, who is supposed to come in at that time. And those are challenges that other teams have faced, that you might want to talk to Team Medics, the senior team. They had problems early on they had bad cells because they were not using the equipment correctly, and they also weren't sticking to their lab schedule, and that set them back about 4 months. So you could learn from their mistakes so you don't replicate them.
And to put it on a more macro scale, there's another team that over a weekend, somebody forgot to feed the fish.
Oh, yeah.
And the fish died. They decided to start over. They came in with bad sushi on Monday morning, and that puts on a bigger scale. But it's the same process. Whatever process you come up with, project management, from who's going to be where, when, who's going to do what when they get there, has to be crystal clear on a collective, common document that you all can see, and all looking at it with however many pairs of eyes are on your team at the same time, and so you all are watching for each other to make sure everything's done. You hate to get to the very end where you have one more step to do, and it gets done twice, or it doesn't get done at all, and messes everything up. So just really, really set yourself up with a system you think is infallible for making sure that everything is done properly.
Make sure you take your lab notes. Team Clot uses a WordPress that they share, so other teams use a Google Doc. Some other teams use a tool for a lab notebook, but whatever you use, you all have to use it. That will be very important.
Mm-hmm.
Yeah, we've already kind of talked about notebooks with Sylvia. So we're going to have one in the lab that just, whenever people come in, they'll sign in what they did. And then another one electronic, where we can sort out the aims and which goes where.
Good.
Okay. Any questions for us?
Actually, I was just wondering. Let's just say, I know for one of the pictures we used, the article was actually from '95. It was from when the cell injury control was first created. If we can't get a hold of the author of the article, should we just not use the picture then, or?
You have to talk to some of the experts for copyright. As I said, images, and we have a copyright person, and I can give you the name of the guy. But images are very, very peculiar. And maybe you can produce your own pictures. By the time you do the lab and everything else, maybe you will have your own picture, so you don't have to rely on someone else's, like the VCU picture. Why would you use, that one probably is not going to be a problem at all, but why would you use that picture? You will use your own lab picture. And maybe you will have your own ones to use. But I can contact and let you know who the person is and how to go about.
Okay, thank you.
And if you have an author that you can't get in touch with, they publish it somewhere, so the journal exists. There's a management board for that journal, and the board will probably give you permission to use the image.
Yeah, I don't think that there'll be a problem. I don't see that, but you have to ask.
Yeah.
And usually it says with permission. It's not only the source, but reproduced with permission. That means that you have received the permission to reproduce it.
Yeah, each source is gonna be different. I mean, authors typically don't hold the copyright. It's the publisher who does. So you don't ask the author, you ask the publisher, basically. And that's about it, right?
Yeah.
In most scientific journals, the policy is that if you are just reproducing for educational purposes, or in a book chapter or something like that, or a review paper, you can use it. Typically, it's already written in their rules, and you don't have to ask the copyright office anything. But you have to figure out that that's the case. Otherwise, yes, you need a permission.
And as I said, for presentations like this, it doesn't matter. I mean, this is for educational purposes, but the final product, as I said, will be published.
That's a good point. I didn't even realize. I mean, I know that, but I didn't realize, because we're not in the time of, of writing the thesis.
Yeah, I know, but
it's a very
that's why
important point to
I
have.
wanted to make sure that
Yeah,
it's something that needs to be
yeah,
considered.
definitely.
All right. But go home with the take-home message that your proposal is a very good shape.
Yeah, and I really liked, I forgot to mention, I really liked the glossary. That part was really, it helped me a lot, but I felt not all the terms were added, like the BBB wasn't there, or some others. So if you can expand that glossary, that will be great for people like me who don't.
And I love the embedded images, with permission or not, I love it. All right, good job.
Thank you.
Good job, yeah.